we are discussing some precise techniques which are used in the advanced molecular biology for diagnosing the different virology concentration, sample concentration, and presence of the viruses in the sample. Now, in this context, the most important technique is immunocapture polymerase chain reaction. That is there, the principle is that we uses a antigen antibody reaction, and then we mix with it the PCR so that we can determine precisely the amount of viruses which are present in the non-biological samples. Now, immunocapture PCR technique is a hybrid method. As I have described earlier, this is a hybrid method because it uses the specificity of antibodies to capture the antigen from the sample, and then it implies the powerful diagnostic power of PCR, that is the polymerase chain reaction, to provide a result which is very, very significant and very reliable. Now, the specificity of PCR, we all know that this method is particularly of use in the diagnostic virology, where the technique allows the capture of viruses from test samples and subsequent diagnosis by the PCR. So, the sample, the viruses from the test samples, they are separated, they are identified by antigen and the antibody reaction, and then diagnosed by the PCR. Now, the technique is extremely useful where the levels of viruses are very, very low in the non-biological sample, such as water or, say, soil or other material which is non-biological, and we want to know that how much viruses are there present in the environment. Now, we can use the PCR microtiter plates or the tubes, and the technique can be carried out in the standard PCR microtiter plates and the tubes which are frequently used during the sample. Now, for example, now we can do that the plate, it is coated with the antibody, then sample is added, and antigen, it captures that antibody there. Then further sample is lysed to release the DNA or the nucleic acid, and then that nucleic acid can be enhanced, can be increased in quantity by the polymerase chain reaction using the specific primers. So this is, in nutshell, is the principle of this technique. Now, what we do, that after washing to remove the excess sample material from PCR reagents, they are added, and the PCR reagent are used to remove the excess material which is there on the end in the titer plates. And then thermocycling, or the PCR, is carried out on the bound viral nucleic acid material. That is the nucleic acid material which is bound there, as I have illustrated earlier in the diagram, and then that is further used. Now, the bound material, then it is subjected to the reverse transcription step, and the RNA viruses will require an additional reverse transcription step prior to PCR. What does it mean is that if we are getting the ribonucleic acid, then we will have to use the reverse transcription step, and that we will have to use the different reagents, and the reverse transcription step would be carried out in prior to performing the PCR. So it means that this technique, as a matter of fact, provides us a handle to determine the low quantity of viruses which are present in the non-biological samples, just as water sample, soil sample, or other material which are non-biological in the environment. So it is very helpful to control and to study the environmental pollution which is there. So we see that this technique has a practical application in the environmental determination, in the environmental control, and in the environmental study. Previously, you see, we can study the biological samples for the virology or the viruses, which is a straightforward technique. But in this case, we will have to use this technique, which is a hybrid technique for the PCR.